Hi, I'm Max Rempel, and I'll talk about the DNA resonance and imperfection as a life force. You can see a bacteria and a white blood cell that is hunting for this bacteria and trying to catch it. As you can see, the white blood cell doesn't have the head, the brain, the neurons. It's just a single cell, and it can do a pretty complex behavior. Here are immune T cells from our blood actively moving around. The idea of morphogenesis and biofield and morphogenetic field comes in part from the study of embryo development, from embryology and biological development. So there is a single cell, fertilized single cell. Somehow it grows into an embryo, and then this embryo grows into a body. And let's do an adult body. And then the adult body keeps the shape for tens of years and reproduces that shape for a long time, for the lifetime. And for dinosaur, let's take a dinosaur, which can be like a large shape. This is like 30 meters, and the cells on the tail and the cells in the middle, the cells in the head, all should coordinate their growth and keep the shape of the body. So the idea was proposed that there is a morphogenetic field which surrounds the body, which is produced by the body and tells each cell what to do. Essentially, the idea is that there are two-directional communication between the cells. They produce the field and they receive the field. So there is a field which is produced as interference pattern of multiple cells, and each is affected by that field, morphogenetic field. The idea of biofield is very old. In yoga, there is a prana energy. In traditional Chinese medicine, it's qi energy. The meridians of traditional Chinese medicine are called nadis in Indian culture, and there are also chakras in Indian culture. In the modern times, Franz Mesmer in 1774 in France discovered the force which he called animal magnetism, and he used the energy from his hands to treat patients. Benjamin Franklin was heading the committee that reviewed that claim, and they turned it down, saying that the results of the treatment were due to the placebo effect. And possibly this was the first time the placebo effect was described. In 1810, Berzelius developed the theory of vitalism, that life is driven by a vital force. In 1840, Johannes Peter Müller said that the presence of a soul makes each organism an indivisible whole, and that light and sound waves show that living organisms possess life energy unexplainable by physical laws. In 1892, Hans Drake introduced the term biofield, and he used developmental biology and embryology based on fertilized sea urchin eggs to study the biofield experimentally. In the early 1920s, Alexander Gurvich, Hans Spemann, and Paul Alfred Weiss independently proposed the morphogenetic fields. Now we temporarily deviate from the idea of biofield to the discovery of double helix in 1953 by Francis Crick, Jim Watson, Rosalind Franklin, and Maurice Wilkins. And later, Jim Watson headed Cold Spring Harbor Lab after Albert Szent-Györgyi, who studied bioelectricity and biologically active water. And soon these two topics would merge. In 1963, Swedish scientist Per-Olof Lodin introduced the term quantum genetics, introduced the idea of proton tunneling in DNA, introduced the idea of delocalized pi electrons in DNA. All three of them I'm actively using. And he didn't expand to whole body biofield, but only discussed the quantum events on nanoscale. And these works were classified secret for 50 years and declassified after 50 years time. In 1969, Stanford Goldman introduced the ideas of biological quantum mechanics and DNA hologram from an electrical engineering view. And this work also was classified for over 50 years. Over 50 years ago, our collaborator on this project, Dr. Richard Allenmiller, in 1972, he presented the idea at the conference presentation. And he developed the DNA hologram as a biofield idea to a pretty much modern state. It was very nice formulated and has most of the ideas which we know at the moment. This work was immediately arrested, classified, and he lately published it in different ways. I'll talk a little bit about terminology. So the classical concept is material. DNA as a chemical substance makes chemical substance RNA, which makes chemical substances proteins, and together they comprise a material body. And in traditional materialistic worldview, that is sufficient, and the body then uses interaction between proteins and uses morphogenetic proteins, they're called morphogenetic proteins, to regulate gene expression, and this way the gradients of proteins are defining the body shape. That's classics. And from the old times, there is another line of thought, that there is another idea that there is a biofield component of regulation where the body produces the biofield, and then this biofield regulates the genes and tells them how to and which position what to do. The holographic idea is coming from linear DNA, it's a linear sequence, into a three-dimensional body. So this is from one dimension to three dimensions, from a linear DNA to three-dimensional body. And the idea of morphogenetic field is similar to biofield, except morphogenesis is specifically creating the shape of the body. So morphogenetic component of the biofield is the one which is responsible for the shape of the body, while other components would be responsible for keeping that shape and then for consciousness and for regulation of different processes. It's important to realize that the DNA in a cell is not naked. In multicellular organisms, the DNA is wrapped around the nucleosome. It is almost 2 turns around a histone core. This is very similar to what a snake ring around a napkin looks like. And this is also important to understand that DNA is double helical, and so this is a double helix which is wrapped around the nucleosome, about a little bit less than 2 turns around the nucleosome. The DNA double helix has a perfect structure, and the nucleosome has also a perfect structure. It's a complicated crystal. And what you see is a tetranucleosome. There is one nucleosome, another nucleosome, third nucleosome, and the third nucleosome here. And this is the largest orderly structure of DNA. After that, the nucleosomes are, the DNA and tetranucleosomes are not arranged in any orderly order. They are quite random. Um, 20 years ago, in 2003, I already spoke about DNA resonance. I noticed that nucleosomes look like electric magnets, and by that time it was already known that DNA works as an electric conductor and even possibly a superconductor. So if the alternated current would go around the through the DNA around the nucleosomes would become antennas emitting electromagnetic waves. And if those nucleosomes would have identical oscillation properties, natural frequencies, then they would resonate. So then I would define the DNA resonator as a sequence that has a specific oscillation pattern or oscillation frequency. And when two resonators communicate to each other through waves, then they would become, they would develop resonance and would exchange information. So oscillation of one resonator would increase the oscillation of another resonator. And then I spoke that there is a need for energy to transmit electromagnetic waves. So some biochemical energy would be spent by one cell, and then it would send out the signal. And another cell with similar resonators, oscillators, would receive the signal. It would spend also energy to receive the signal and then transform it into a biological or biochemical signal. For example, it would express a gene. The DNA would express a gene or transcribed into RNA. In addition to electromagnetic wave signaling, we stumbled upon the idea of crystal propagation in water. So DNA signaling through electromagnetic waves implies the waves, which are patterns in time. When we talk about crystal propagation in water, water structuring by DNA, these are patterns in space. So we came upon the old idea of others that DNA may initiate microcrystallization of water and imprint its sequence, the DNA sequence, on water structure. So we call this crystal pattern propagation or snowflake signaling. In snowflake, the pattern is propagated in multiple directions. And this deserves further study because the patterns are changing depending on the distance from the center. The patterns are changing. So that's I think is a very unexplainable phenomenon which needs explanation. So in biology, microtubules are known to crystallize, self-crystallize from monomers. So if there is a lot of monomers, the microtubules grow. And when the monomers are sparse, the microtubules shrink. So that's a dynamic self-regulating process, growth and dissolution of the microtubules. So we suggested that in the nucleus, DNA also propagates its pattern, imprints its pattern on water, and this pattern propagates outside of the DNA as crystal structures. So the cell contains the outer wall, cell wall, and the nucleus has nuclear wall. And inside the nucleus there are chromosomes, and the liquid in the nucleus is called nucleoplasm. And between the nuclear wall, outside of the nucleus, and within the cellular wall, there is a cytoplasm. So we propose that there are chunks of crystal water like snowflakes or ice slur within a nucleoplasm. And it's very important for our model to understand that it is a very thick, heavy fluid nucleoplasm containing a lot of DNA. So as I said, our body contains about 2 or 300 grams of DNA. And in the nucleoplasm, DNA is about 1%, which is a lot because it's a very long polymer, which makes it very thick when it's not condensed in the interface. And it contains about total DNA and proteins contain about 19%, so about 18% of the proteins and a few more other components, lower molecular weight. And so the water comprises only about 78% of the nucleoplasm. So it's a very thick moving gel. So this video shows that chromatin is actively moving. So this is the nucleus of the cell. And so that's the nucleus of the cell, zoomed in. And that's a real video. And you can see that in the interface where the cells are not dividing, the cell is actually actively working, and the green and red chromatin are just two different types of chromatin. So chromosomes are unwinded and untied, and they fill up the whole nucleus, and they are actively moving. And this video is sped up about 100-fold, but still it's a very fast process of moving within the nucleus. And I was trying to find a real-life example from the macro world of self-organizing gel, and one of the good illustrations is an octopus, how it moves. It's like a gel which is self-organized, and it can move in a very sophisticated ways. So this is an example of how I see the movement of chromatin within the nucleus. And here is a very good computer simulation of how I see biofield. So that's some sort of invisible field which also moves around and is an living interference pattern, self-organizing pattern. So it's important to separate a biofield from the physical material movement. So there is a physical material cell, and within the cell there is a physical material nucleus, and there is material DNA there, and there are clusters of water also moving very physically. This is physical thick gel moving, and on top of it there is invisible electromagnetic or subtle fields which also are self-organizing. And the best interaction between these subtle fields and the material gel is when this material gel is also moving and self-organizing, and there is a lot of crystallization and dissolution processes. So basically, when there is a lot of choices being made, that's where subtle influences of the field are most pronounced. So this is the end of the introduction. So far I introduced the ideas of DNA hologram, a morphogenetic field, nucleoplasm as a self-organizing gel, nucleoplasm as continuously growing and dissolving suspension of microcrystals of water, and the idea of chromatin, the substance of chromosomes, being distributed over the nucleus and being self-organizing and continuously self-organizing. Next, I will talk about a molecular model which we have built, and the computational testing of this model using real data. So the hexagonal nature of the classical ice, which is called ice 4, is well established. It's a honeycomb which consists of layers of water, and they are hexagonal. These are real microphotographs obtained with atomic force microscopy. So here is DNA double helix. And this is a classical base pair. As you know, the DNA letters are G and is paired to C, and A is paired to T. So we noticed it a long time ago. We noticed that the base pairs of DNA contain also hexagons. So here is a base pair, G, C, and we fit it in a honeycomb hexagonal structure of water, and the proportions are approximately correct. So here is a double helix of DNA, and the base pairs, as you know, are perpendicular to the double helix of the DNA. And we just expanded each base pair and proposed that the layers of water hexagonal will be built around the base pairs and expand outwards, and that the layers of water will be perpendicular to the main axis of the DNA. Here we built a very preliminary imprecise model of only two steps of the DNA ladder. There is one base pair here, there is one base pair on the bottom, there is second base pair on the top, and there is a layer of water which is coplanar with the same plane as the first base of water. So the bottom base of water is expanded in a hexagonal honeycomb structure of water. And all the other parts are not shown. The second layer is not shown, and the DNA continues in both directions. So the DNA goes up and down, first strand, another strand. So the classical model of ice didn't work very well for our model. Essentially, in classical ice, the layers are not flat, and moreover, they are bound to each other via hydrogen bonds very strongly. So they cannot shift relative to each other. The shift is prohibited. And then we found the model of Lippincott published in 1969. It was called polywater. There were a few years when polywater was discovered, and it was researched in many labs around the world. But then, for political reasons, around 1972, it was excluded, became prohibited, and there was no more publications. So this polywater is attractive because the layers of it are flat, and the hydrogen bonds between the layers are weak. And this allows the layers to shift relative to each other in multiple directions. I will show that on the next slide. And that allows to imprint biological information on this layer. So this is a more likely structure to be responsible for life. So that's a structure of, we hypothesize that this is a structure of the living water within the nuclei in the nucleoplasm. So this polywater is hexagonal. It contains incomplete hexagons. This is complete hexagon, but most of them are incomplete hexagons. The first interesting feature of polywater is that it is imperfect. It cannot form the complete honeycomb. It contains a lot of space for single water molecules, for like actually liquid water. But as it grows, it accumulates negative charge. So if it grows too far in one direction, it will start dissolving. It would be beneficial for it energetically to start dissolving to reduce this negative charge. So it would grow on one end and dissolve on another end, or grow on one end and dissolve also on the same end. The next interesting property of the polywater is that here is the first layer, and here is the second layer. And the layers are shifted, always shifted in one of the 6 directions. And this allows for the biological information to be recorded in the multiple layers of polywater. If you remember, a few slides ago I showed a double helix of DNA and the layers, planes of polywater perpendicular to the DNA. And the size estimate suggests that for every one nucleotide, for every one base pair, there will be 2 layers of water. Another nucleotide, another 2 layers. So they are twice as frequent. So if you consider 2 nucleotides, there will be the first layer of water, intermediate layer of water, and the third layer of water. Then it's helpful to estimate how the layers will be shifted, how the third layer will be shifted relative to the first layer. So the first nucleotide, the second nucleotide, and the first layer of water and third layer of water. So as you saw previously, the second layer is shifted in 6 directions. So these are the 6 directions. And the next step, it can move backward, it can move forward, it can move this way, it can move this way, coming back to the position of the second layer, move this way and this way. And if you do all possible movements, there will be 12 points on outside, 6 points on inside, and a center point. So 19 positions total would be the position of the third layer compared to the first layer. And angles, there will be 12 angles of movement. They move in parallel, so the layers shift in parallel, but the position will be angled in 12 angles. 1, 2, 3, 12. And here it is how we propose that DNA imprints its sequence on the water. So the letters can be positioned in 2 ways in DNA double helix. First way is when the left strand of the double helix has one ring. This is either letter T or C. And this is a double ring. The letter would be G or A. And the second orientation is when the double ring, 2 rings of water, double ring, it's called purine. The purines with 2 rings are on the left, so G and A would be on the left, and single ring C or T would be on the right. And this is actual orientation in the double helix. And now we can see that in these 2 orientations, double ring on the right and double ring on the left, we will have different positions of the hexagon. So if water honeycomb is built from hexagons of DNA, then like light brown honeycomb would be built for the first structure. That structure fits light brown honeycomb, the first upper structure where the purines are on the right and pyrimidine, the single ring, is on the left. And in the second orientation, when purine is on the left and second ring on the right, the green honeycomb would be built around the base pair. So this way, the structure of the DNA would imprint on the water structure. Here I expand the idea of DNA sequence imprinting on water structure from a single base pair to multiple base pairs. So here is a picture from previous slide. the water layers shift depending on the position of the purine, and then you can see in this picture pyrimidine on the right, purine on the left, and this is pyrimidine C, and this is G. And then the next one is purine on the right, and this one is A, and this one is pyrimidine C, single ring. And you can see that water layers can shift, and you remember from previous slide, they can shift, one of the previous slides, they can shift in 3 directions. And here is the idea that a sequence of a double helix would shift water layers. Here, like I have a flat view basically, but it's possible to imagine that water layers will be shifted in 3 directions. And next idea is that if you have 2 DNA double helices, 2 pieces of DNA near each other, they could form a common structure through the water, and they could coordinate their sequences. So if the DNA have similar 2 DNA double helices, if two double-stranded fragments are near each other and of identical sequence, they would form a perfect water structure, and the shifts in water layers would synchronize between the two. And if we consider that there are intermediate layers between this, for every nucleotide we'll get 2 layers of water, then the movement goes in 6 directions of the first layer and 12 directions for the next layer, and total will be 19 positions for the second layer of water. You're already familiar with the nucleosomes. So in multicellular organisms, DNA is packed and wrapped around the nucleosomes. So there is it's called beads on the string structure. DNA is wrapped around the nucleosomes. And this is a double helix of DNA. Double helix of DNA. So 2 double helices sit next to each other. As you saw in the previous picture, the hypothesis was if two double strands sit next to each other, then they could form layers of water which would bind them together, and they can coordinate the sequences. So if the sequences are identical in purine structure, then they would form a more perfect structure. So here is the idea. So here is purine, pyrimidine, purine, pyrimidine A, T, G, C. And the idea is that, and here is the same structure, just zoomed in. So this is the same structure from here, this one. So the idea is that shifting of the sequence-dependent shifting of water layers would be synchronized between the 2 sequences. Obviously, that can happen during the evolution. If that was beneficial for their structure, then evolution would synchronize the 2 strands, the two strands in the nucleosome. So these 2 strands would be sequences in their purine sequences would be synchronized. And that's what we can test using available genomic sequence. 20 years ago, we suggested that a nucleosome is an instrument, the tool that allows the nature to read the DNA sequence and to convert it in electromagnetic waves. Now we propose that in addition to that nucleosome is the unit which reads the DNA sequence and converts it to water structure. And now we are looking specifically which DNA sequences are involved in that. So this is just another view of nucleosome, a 3D view. You can see 2 double helices, like 1, And this is the same structure. And now we unwrap it. And this unwrapped, this part of the loop, number 1, is this number 1. And this part of the loop, number 2, the second loop is number 2, loop number 2. If you unwrap it, and they look at the same direction, they are oriented in the same direction in the chromosome sequence. And the size of the nucleosome is pretty fixed, and it's about 77 base pairs, nucleotides, which is the same thing as nucleotides. So these are 2 loops of nucleosome, 1 and 2, and this is 1 and 2 loops. And they're facing the same direction on the chromosomal sequence. And the length of those loops will be roughly estimated to be 40 base pairs, nucleotides, 40. And if there are 2 repetitive sequences, then they're called tandem repeats. But if there is one repeat, then there is a gap, and then there is another repeat, then it doesn't have a name, so we call it jump. So it's a jumped sequence. So we study those jumps. For that, we take the whole human genome. We took the whole human genome, 3 billion base pairs. Programmatically took only intergenic regions, 45% of that. From that, took only the fragments bound by nucleosomes, which is about 20% of that, of the previous. From that, took only the fragments which are conserved in evolution, which is 20% of the previous. So these are functional, bound by nucleosomes, important parts of the genome. And we looked for this purine jump. So these purine jumps defined as homology, pretty much identical, but not 100%. Not 100% identical, somewhat similar sequences are located at a specific distance. And we took the distances from 40 to 140 base pairs. So our sequence is, our jump length is 77, so in the nucleosome 77. And we took the range from 40 to 140. And then we divided the obtained jumps by the length of 50 to 58% homology, 58 to 67% homology, 67 to 100% homology, roughly about the same number of hits. And then we plotted, so horizontal axis is the size of the jump. So 77 is right here, 77 jump. Length 77, this is 140, this is 40, and the vertical axis is counts of how many times this jump happened in the genome, in a subset of the genome that we picked. Basically, most interested, most promising subset. And we obtained this peak, which is around 77, which is what we sought. And there are other peaks also here, and other peaks here. And I summarize them here: 56, 66, 77, 92, 105. So in the nucleosome, this peak 77 corresponds to classical nucleosome, this one. And the smaller jumps would correspond to smaller nucleosomes. So there is like possibly there exist nucleosomes of smaller size, bigger size, classical size, and even bigger sizes. So as you can see, we received this peak at 77 nucleotides, and this corresponds to that distance. And initially we built that model and produced that data. So this data is confirmed that the model is likely correct. And this model was built on the idea that the shifts between the water layers are produced when water layers are built upon the DNA basis. So depending on DNA sequence, the water layers will be shifted. And the structure of shifts in water layers is based on the structure that Lippincott 1969, which is has the incomplete honeycomb with hydrogens in the same plane and oxygens, and the only way this structure can exist is when the layers are shifted in 3 directions, one of the 3 directions, each layer next to each other, next to the previous layer. So this is the evidence that the model is possibly correct. An additional consequence from this is that their double helices, 2 double helices, might interact with each other through shifting of the water layers. And this shifting might, these water layer structures can possibly expand through the whole nucleus. So the whole nucleus can contain the water layers which have DNA sequences imprinted on them and define the patterns of these structured water layers, clusters of water. Next, I started building the structure of water surrounding the DNA, and the water has it's called crystallographic symmetry, so it reproduces itself if you turn it 1/6 of a turn. Basically, it's symmetrical with every 1/6 of a turn. It reproduces itself. The honeycomb. The DNA is much more complex, and its basic symmetry is it reproduces every 2 turns it reproduces itself, and it has 21 steps per 2 turns. So this is the ancient medieval staircase, double helical, and it has about 14 steps per 1 turn of the double helix. And in DNA it is 10.5 steps per turn, or 21 steps per 2 turns, and then the DNA actually comes to the structure which is pretty much the original position. So this is the classical, the hexagonal honeycomb, and this is the DNA classical B form, and it has 10.5 steps per turn, or 21 steps per double turn. And it doesn't fit well into the honeycomb. So we try to build that structure, and the classical B form of DNA, which is mostly present in the living cell, only aligns poorly, like maybe step number 2 aligns a little bit, step number 3, 4, 5 aligns a little bit, 7 aligns a little bit, 8 aligns a little bit, but in general it's a very poor alignment. So this DNA is twisted. If we untwist it a little bit, then to the level, so this is 10.5 steps per turn. If we untwist it to 12 steps per turn, then it aligns pretty well with the honeycomb. So yellow is red one is the honeycomb, and blue one is the double helix, and the steps are 12 steps per turn. So from 0 to 11 is 12 steps. So what we see here is that steps 0, 2, 4, 6, 8, 10 are perfectly aligned with the honeycomb. It's a perfect orientation, perfect alignment. And the opposite is like even steps, even are good, odd are poorly aligned, they're misaligned. So this structure has very interesting consequences. So for the even steps, suppose this is like 0, 2, 4, letter 6, 8, 10. So for these letters, alignment would be fine, and the two strands, two double helices, would communicate through that layer of water because they align well. The next layer of water will be misaligned, and there will be poor communication between double helixes. The next layer will be good alignment. The next layer will be bad alignment, bad, 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 and good, good, good. So even would be good, odd would be bad, or the other way around, odd would be good or even would be bad. They are pretty equal. So one phase would be aligned and another phase will be misaligned. So that predicts that only every second letter in the code would be aligned through the purine homology. So I call it even purine homology, and I will explain what I mean in the next slide. So this is a picture from the previous slide. Their hexagon is the honeycomb of water. The blue one is the DNA, untwisted to 12 steps per turn, and even nucleotides are perfectly aligned with the honeycomb. Odd nucleotides are perfectly misaligned with honeycomb. And here I give the example of two sequences which are completely dissimilar. I intentionally designed them to have different sequences. And this is the sequence number 1. It is the same sequence number 1, same sequence number 2, number 2. And the numbers here correspond to the positions in a double helix. So 0 would be this one, 1 would be this one, 2 would be this one, 3 would be that, and so on. So these are the DNA positions in the double helix, and these are two double helices. So this is sequence number 1. That's it. That's it. This is sequence number 2. That's number 2. Number 2. And they align or become. They are communicating only through even layers of water. So this is the layer where they are communicating 0, 0, 2, 4, 6, and so on, and so on. So 0, 2, 4, 6, 8. So the purple color here, the purple is purines. The purines, as you know, are purines are labeled purple, and they have double rings in the DNA, and it's A and G are purines, and pyrimidines have single ring hexagon, and they're T and C, and they're labeled actually white background. So these are purines, and these are the in between of them is pyrimidines, white. So if I block every second letter, like I put here a comb and just block every second letter, so only even letters are left, 0, 2, 4, and so on. Then there is a perfect agreement between the two sequences, sequence number 1 and 2. So there is like purine, that's purine code, pyrimidine, 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 same color, pyrimidine, pyrimidine, purine, purine, purple, white, purple, purple, purple. So these sequences are different, but they agree in a even purine code, so I call it even purine. Basically, if you take only even letters and you look only at purine code, they are identical. So how does it correspond to this picture? So the sequence number 1 is this one, number 1, this one, number 1, number 1. Sequence number 2 is this one, number 2. And we only pay attention to every even zero. So 0 is here. They agree. So these two double helices, they agree on the first letter. The second, they disagree. We cross it out. Cross it out. Then the next one, they agree again. They agree, disagree, so on. So only even letters would agree, and only odd letters would disagree. So the two sequences would correlate to each other, would communicate to each other through water letters, through water layers. They would only through even code and only through purine code. Now we take the same principle. So that's even purine principle, we take it, and we apply it to two loops on the nucleosome. And these two loops would correspond. This would be loop number 1, this would be loop number 2. And these are like number 1, number 2. And if we unwrap the nucleosome, like nucleosome, we unwrap it, and this would be number 1, number 2 loops. But we pay attention now only to purines, and we ignore only to even nucleotides, and we ignore the odd nucleotides. So only every other nucleotide we pay attention to. So now we took the same 3 data sets from the previous analysis, and we required that only homologous, that even purine homology, basically that only even nucleotides should have certain level of homology. I think we did 80% homology. So we picked these ones, and this whatever homology was, we set it as 100%. And we look at odd nucleotides, what homology would be there. And this would be the homology in the same pair, would be the homology of the odd nucleotides. So these are even, and this would be odd. And the homology was way lower. It was about 25 to 30% lower. And this was reproduced in 3 different subsets of data, and this is a very significant result. So essentially that confirms that there is a preferential homology in even nucleotides and preferential dissimilarity in odd nucleotides. This result was produced only on human genome, and we are intending to test this on a mouse genome, and it was done using one programmer and one program, and we would like to make sure it works with a different programmer on a different genome. And also it was tested only in cis position, where both fragments are located in the same double helical DNA strand, and we want to test whether this homology, even purine homology, also is significant between the strands which are on different chromosomes or very far apart from each other. And this was cis, and this would be trans. If it is true, then it would be a pretty significant. The meaning and impact of this result would be pretty significant. To explain the significance of potential even purine homology, here show again the slide that you have seen before. It is cell nucleus. So there is a cell, there is a nucleus, and that's a cell nucleus, and the chromatin, the substance of chromosomes is fluorescently colored. And this shows that chromatin is constantly moving. Basically, when the cells are not dividing, they are in the interphase phase of the cell cycle, and chromatin is constantly moving. And this video is sped up many times, but still it is very important that chromatin is very dynamic. Here I remind you again what even purine homology is. So essentially, out of the sequence A, say T, C, G, T, we only take even nucleotides or odd nucleotides, doesn't really matter, one of the two phases. So we hide all others, we take only even nucleotides, for example, and then we look only at the colors between two different sequences. And the color here signifies the if it is purine or pyrimidine. So pink ones are purines, and white ones are pyrimidines. A and G are purines, C and T are pyrimidines. So in different sequences, if we take only even nucleotides and then they're similar in their color, that is in their purine pyrimidine property, then it's called even purine homology. And it can be 100% homology or it can be 80% homology, but it's even purine homology. Now let's look at the two double helices. Essentially, we claim that it is double helical DNAs through water become coordinated. They attach to each other and form with water a coherent crystalline structure. So there we look at one base pair, and this would be even base pair and like number 0, and then there is a homology there will be agreement, and then the next base pair would be skipped because it's misaligned, the angle is wrong, and it wouldn't form a good structure with water. And then next one will be again perfectly aligned, two base pairs, and then again skipped, then next again, and so on. So this is two double helices. And if that works in cis position, that would be like when the two sequences are in the same double helical strand of DNA, that's cis position, cis. And if it is between the two strands from different chromosomes or far apart, like a big loop, then it's called trans. So if there is that homology, alignment. So the idea is that this is like a big strand of DNA, big double helical strand of DNA, double strand. And then when it comes together, it would form homological alignments and would kind of stick to each other. It will be sticky. If using the principle of even period homology, this one. And that would explain how chromatin is constantly rearranging in a sequence-specific manner. And then we claim, and that's pretty confident claim, that rearranging chromatin, chromatin dynamics, works as a bicomputer within each cell. Essentially, it is how the genomic sequence, the sequence of the genome, rearranges itself, and that's how it thinks in the cell, in the nucleus. Here I want to do one more step. So you have seen the image of the nucleus and chromatin dynamics in the nucleus, and the suggestion that the double helical DNA is connecting to each other through regions of homology, which are formed by the principle of even purine homology, regions of homology. And if we zoom in into that region of homology, we get, on the previous slide, we got two double helices connected through even purine homology. Then I zoom in into the two double helices, and I get this structure just the same. And what is notable about this structure is that DNA here is straight and naked. And in our cells, in eukaryotes, with the cells which contain a nucleus, DNA, as you know, is wrapped around nucleosomes. So straight parts of the cells of the DNA are pretty rare. So straight parts are pretty rare. So the straight parts are short, and mostly it is nucleosomes. So then I have to come up with the idea how that could work with two different chromosomes, or distant parts of the chromosomes. How can they come together and form a structure, form a point of contact through water, through the layers of water, and keeping this even purine homology? And that is tough. I don't have the answer yet, because when you put the honeycomb structures flat on top of each other and they are parallel, that's a pretty easy to imagine picture. They form pretty nice layered cake or crystal, which is pretty standard. Now, if you have to connect the nucleosomes, then it has to be like cylindrical structure, and multiple cylinders fit into one another, and that is much harder because it's just harder. It does look pretty. There is not many crystals that form that structure, cylindrical. And even more, if we could try to build a helical structure or a vortex, then it becomes even harder. So that is an open question. I don't have the answer for that yet. I will explain this question just a bit more. So suppose you have a classical crystal. Then the spacing between molecules, monomers in the crystal, usually is equal. And that's a typical crystal where you have spacing equal, and then everything is parallel and it can continue in any direction, grow in any direction, and z direction. Then if you take a cylinder, suppose you have spacing in a cylinder, let's do 6 for example, then when it goes radially, then it's the spacing will become larger and larger, so the molecular distances wouldn't be real. So it has to go it has to grow in some pattern, which is hard to imagine how it would happen. Possibly there are some solutions, but I didn't research it. I'm not aware of cylindrically growing crystals. Next, so Anirban Bandyapadhyay predicts that water structures around DNA would be forming helixes and vortices. So a vortex is a helix which comes from a wide diameter, comes to a point, and it might come out of the point. Essentially, the structure of it is coming to a point and then going out of a point. And it has, again, a problem of, it's solvable problem, but it has to be solved, that their distance between monomers has to increase somehow. And that's hard to, it's not allowed by traditional chemistry. So that requires a fractal crystal or fractal vortex, and I know one fractal in water, which is a snowflake. Essentially, it's a basic structure of hexagonal snowflake, and then six-ended snowflake, and then a part of it is again producing a a smaller snowflake, and a part of it is producing a smaller snowflake, and a part of it producing a smaller. So that's a fractal. How that translates into fractals around DNA, I don't know. And the second problem is, how does it reflect DNA sequence? For me, the sequence is very important. It has to imprint on water. That I don't know. It requires additional research. Now, for the cylindrical water structure, I can imagine a small cylindrical water structure which is made out of hexagon. So a hexagon laying on its side. So we're looking from it from a side view, and then we start expanding it that angle has to be 120 degrees, 120. And then we expanding it by twisting it a little bit this way, and it goes up and up and up and up and up. And essentially, that is a possible structure, but how would it be applied to structure around the DNA? That requires more thinking. Now we come back to the parallel water layers made out of honeycomb water structure, and these layers being perpendicular to the axis of the double helix. And look in more detail how these layers can be built. There are two flat structures of water where the hydrogens are not sticking out of the plane and are in the plane of water. And this allows for the multiple planes to be crystallized over each other and shift. So this way they would reflect the structure of DNA, purine structure of DNA. So these are two structures. One was published by Lippincott, and another was published by Pollock. Lippincott 69, Pollock 2014. The problem with Pollock structure is that it's heavily negatively charged. And Pollock shows that easy water that he studies is negatively charged, but this negative charge, where you have one negative charge per water hexagon, is too huge, too big. It doesn't make a lot of sense. While Pollock's structure can incorporate negative charges like this hexagon, but sparsely. Essentially, it's imperfect structure with a lot of imperfections, but it is flat, it is honeycomb, and it is mixed with water molecules which are outside of the crystal structure, and it is continuously growing and dissolving in water. So this is pretty much a snowflake, just a flat honeycomb snowflake that continuously reorganizes and recrystallizes. This makes much more sense, and that's what we used. Here I show how their Lippincott structure grows. So it has liquid water filling the canyons, the gaps between the branches. And it crystallizes like a snowflake, and it's flat, like a snowflake. And as in water, the turn is 120 degrees. So oxygen is in the middle, hydrogens are on the sides, and the turn is 120 degrees. And if you, at every step, there should be a turn. So oxygen, hydrogen, straight oxygen, and at oxygen there will be another turn 120 degrees. 120, 120, hydrogen, straight 120, and it can be turned any direction. And if you just follow these rules, you will build a honeycomb structure. What is permitted is, so the charges. The first hexagon is negatively charged, but as it has branches, it is neutralized, so this structure is already neutral. It's neutralized, so slight positive and one negative charge, one negative. And then the branches can grow as long as possible, and they can branch also. They can branch as much as possible, like in a snowflake. They can branch. And what is prohibited is that merging of the branches. So two branches, when they merge, another negative charge is formed. When they form a hexagon, another negative charge is formed. So this makes the structure less stable. So the branches can grow but cannot merge, and they can branch but they cannot merge back. And I don't know why, but possibly there is a principle of keeping the structure compact. So they kind of don't go very far, but they kind of circle back and keep the structure compact. That is a possibility. So now I'll show how it grows. So first is hexagon with negative charge, and then the branches grow, and grow, and they rarely merge, so the overall charge is neutral. And then when the hexagon are formed, they are charging negatively. They're becoming negatively charged. And when the negative charge grows too big, then the structure would start dissolving because it becomes unstable from too much negative charge. So it's a dynamic structure with canyons of liquid water and a crystal that continuously grows or dissolves, grows and dissolves continuously. And here I illustrate that the structure that accumulated too many hexagons or merged branches has too much of negative charge, and it starts dissolving. So it dissolves, dissolves. I illustrate, this is the side view of the double helix. This is the double helix, the top view, the same double helix, just the top view. And here I show how it initiates the crystallization of water into polywater, and then that grows, the front grows, and then it starts dissolving because it reaches optimal maximum size, and it might continue traveling as a crystal wave. So here is an idea of traveling crystal wave. So DNA can create a crystal which then would propagate through the nucleoplasm and carry the structure of DNA away from DNA. So there could be a wave, and imagine two waves of different structures or similar structures meeting together in interference. So that it will be like similar to interference, but in a crystal. Crystal waves would interfere. So in the top view you have a honeycomb, in the side view you have just flat layers. Here we built a molecular structure of Lipin quad structure, polywater structure. So it's multiple layers, and there are canyons or rivers inside, on the sides and inside, and they're filled with liquid water. And sometimes it would grow, and sometimes simultaneously it could dissolve, and it could travel, or depending on circumstances it grow and dissolves. So it's continuous crystallization and dissolution like in an ice slur. And this is a real picture illustrating the process. You have long-term ice, possibly Arctic ice, and you have river in ice, and depending on the weather it would either crystallize or dissolve, and depending on day and night it would continuously grow and dissolve. So that's a very similar process. Another consequence from polywater structure, from honeycomb structure, is the imperfection of the angle, how DNA fits in it. We already reviewed it in even purine code. Essentially, this is water, and this is DNA, and at best conditions, if there is 24 steps of DNA per 2 turns or 12 steps per 1 turn, if DNA is a little bit untwisted, then it perfectly fits every other base, aligns perfectly with the honeycomb around the DNA, honeycomb of water. So that would be this direction, and so on, and this direction, and that direction, and that direction, and that direction. 6 directions are perfectly aligned, and every other nucleotide, every other step of DNA would be imperfectly aligned, and it would be unhappy, energetically uncomfortable, energetically suboptimal. So the DNA would tend to twist back and forth like this. So DNA would twist back and forth. That's the simplest way to resolve. So first one nucleotide will be aligning with water, and then another one, like one phase and then another. Even odd nucleotides. And while twisting, DNA would screw in the water and screw out. So the oscillation would be back and forth, in and out, in and out, in and out. That's the simplest oscillation. Obviously, it can go further and do multiple twists, but that's the like one step back and forth. So here I summarize permanently unstable dynamic self-reorganization of DNA solutions. First, it is a recrystallization of polywater, which we just reviewed. So liquid water, canyons of liquid water are constantly crystallizing into polywater, and polywater is constantly growing and then dissolving permanently, dissolving and recrystallizing non-stop. Then second is screwing oscillations back and forth of DNA into layers of water because of the angles, hexagonal angles and angles of DNA don't match each other. So DNA would constantly be imperfectly situated in water, and that would cause it to move all the time, which we know happens. And the next one is chromatin dynamics, which is a well-known fact, and we added there sequence-specific, most likely even sequence-specific alignment of two double helices, different parts of chromosomes and of the same chromosome or different chromosomes. They potentially align through water and possibly through even purine principle, even purine code. And this is leading us to the notion or conclusion of imperfection as is the basis of life. So imperfection is the basis of life. So on the previous slide, we reviewed recrystallization of water, screwing oscillations in DNA, and chromatin dynamics. And it's all permanently unstable dynamic reorganization and imperfection as a basis of life. Now, it's important to highlight that continuously unstable states of reorganization are perfectly suitable for subtle effects of the morphogenetic field. And if you remember, morphogenetic field is the field that defines the shape of the body and the structure of the body. Now, the fields can be of electromagnetic nature, including heat, radiant heat, and subtle fields, and acoustic and electroacoustic nature, essentially electromagnetic and mechanical oscillations. Here I illustrate the idea of conversion of electric oscillations into sound and back. So here is a regular speaker, and the electric voltage, alternated voltage, is applied to electric magnet in the speaker, and the electric magnet produces oscillations, mechanical oscillations, and mechanical oscillations produce sound. And the reverse, the microphone works by receiving the mechanical sound oscillations and converting them to alternated voltage, alternated current. Here I illustrate that a nucleosome and DNA can work as a converter, converting electric and electromagnetic oscillations into the sound and back. And the size of the wavelength does not necessarily have to correspond to the size of the object which oscillates. For example, the voice sound has the wavelength of about 2 meters, but it resonates with our ear, which is much smaller. About the eardrum is about half a centimeter. Here I show the conversion table. The frequency is common between the sound and light, or electromagnetic waves. So here is the light wavelength, common frequency for light and sound, and the sound wavelength. And here are typical objects of interest. Nucleosome diameter is about 10 nanometers, correspond to 150 gigahertz, and that would correspond and convert to the sound of, or to the light wavelength of 1.5 millimeter. 42 gigahertz is a typical millimeter wave therapy frequency. That would correspond to 36 nanometer sound wavelength and 5.3 millimeter light wavelength. Mammalian cell would have the size of 20 microns, which would correspond to 75 megahertz, and light wavelength would be 3 meter. And the sound frequency of 192 meters would correspond to Schumann resonance frequency of 783 gigahertz, 7.83 hertz, and that would correspond to 28,000 kilometers, and that would correspond to the sum of the sizes of the measurements of our planet. Here I illustrate that a nucleosome and DNA can work as a converter, converting electric and electromagnetic oscillations into the sound and back. And the size of the wavelength does not necessarily have to correspond to the size of the object which oscillates. For example, the voice sound has the wavelength of about 2 meters, but it resonates with our ear, which is much smaller. About the eardrum is about half a centimeter. Here I show the conversion table. The frequency is common between the sound and light, or electromagnetic waves. So here is the light wavelength, common frequency for light and sound, and the sound wavelength. And here are typical objects of interest. Nucleosome diameter is about 10 nanometers, correspond to 150 gigahertz, and that would correspond and convert to the sound of, or to the light wavelength of 1.5 millimeter. 42 gigahertz is a typical millimeter wave therapy frequency. That would correspond to 36 nanometer sound wavelength and 5.3 millimeter light wavelength. Mammalian cell would have the size of 20 microns, which would correspond to 75 megahertz, and light wavelength would be 3 meter. And the sound frequency of 192 meters would correspond to Schumann resonance frequency of 783 gigahertz, hertz, I'm sorry, 7.83 hertz, and that would correspond to 28,000 kilometers, and that would correspond to the some of the sizes of the measurements of our planet. Here I use the same conversion, electroacoustic conversion for sound wavelengths in red, electromagnetic wavelengths in blue, and frequencies common to sound and electromagnetic spectrum. And here I highlighted the most common known electromagnetic frequencies, including light, ultraviolet, X-rays, gamma rays, Wi-Fi, cell phones, microwaves, radars and radio, and infrared and terahertz radiation, including 5G, which would be somewhere in that range. Now, what is most important for DNA resonance is which of the therapeutic frequencies produce high therapeutic effect at very low power. And one of the most key frequencies is millimeter wave therapy, which is 42 gigahertz. 42 gigahertz. Very interesting, pretty nicely studied therapeutic frequency. Another one is for low-level light therapy, which is around red and near-infrared light, 660 nanometers to 850 nanometers. That's a typical near-infrared therapy. And UVB therapy at low power, UV is actually therapeutic, and that's also a very important frequency. But all others obviously are great. Ultrasound therapy, ultra-high frequency therapy, and pulse electromagnetic field therapy, and TENS. Sound therapy is also very important, poorly studied, but very important. So all of these frequencies are of great interest for DNA resonance. I believe DNA can resonate at multiple levels, at multiple structures, and especially with water, it can host a whole number of resonant frequencies. As you know, my main focus is to figure out how does DNA sequence produce a morphogenetic field or DNA hologram. We know that DNA can serve as a conductor of electricity. It is an insulated conductor, and nucleosome is a magnetic coil. Can become a magnetic coil. And the nucleosome can also run through the DNA, so nucleosome can roll over the DNA, or DNA can slide through the nucleosome. So this way, the nucleosome can read DNA sequence. So DNA sequence can either roll or slide through the nucleosome. And that resembles how the magnetic head of the video player works. So this is magnetic head of the video player, and the tape on it is wrapped around at once. In nucleosome it's wrapped around almost twice, but here it's wrapped once. But there is also, what is funny, it is also a left coil, a left turned coil, and nucleosome is also a left screw, left coil. Since DNA is a conductor and nucleosome resembles a magnetic coil, the current coming through DNA should produce a magnetic moment or magnetic vector, magnetic field. And this is the model of a tetranucleosome, which we published 6 years ago in 2017. And it contains 4 nucleosomes. 1, 2, 3, 4. And that's a very well characterized structure. It is the largest orderly DNA structure. It has a pretty perfect shape, while all bigger structures of DNA are pretty disorderly. The order disappears after that. All larger structures are messy. So in this structure, if the current goes one direction, then the magnetic vector goes this way, and then this way, and then this way, and then this way. So basically we observe the oscillation of electricity and magnetic vectors. Here is the same model, just with more electrons shown how they travel. And also I show how they come together and repel from each other. And repel from each other this way and this way they repel. Negative repels, positive charges I just don't show because there is too much here. This is a hypothetical model, not proven yet. Now let's zoom in and focus on electric and magnetic properties of the DNA at smaller scale. So as you can see, this is a double helix of DNA. And here the outer backbone, and inside there is a base stack. The core of DNA is called base stack. And if I remove the backbone, this is the base stack. It's also double helical, but backbone of the DNA is not shown. And the strands are, this is the green one is one strand, and blue is another strand. And here are electron wires in blue and proton wires in red. Proton wires are much harder to study because very little known about them, and they also include the water surrounding of the DNA. The water comes very close to the base stack. It basically connects very tightly to the backbone, to the charged phosphate backbone, and comes very close to the base stack, but it doesn't penetrate the base stack. So outside of the base stack there is lots of water, and it is a hydrophilic area of DNA. So sugar phosphates are very hydrophilic. Inside the base stack, it's hydrophobic. There is no water. It's very hydrophobic, and that's actually what holds the DNA together. The bases are hydrophobic. They escape water and pull together and are attached together, while the negatively charged backbone tries to linearize, but because the bases are pulling DNA together and the backbone is trying to separate it, that forms a perfect double helical structure. As you remember, purines have 2 rings and pyrimidines have 1 ring. And here we can see that 1-ringed pyrimidines produce a single electron wire, and 2 rings purines produce a double electron wire. It's a double. So depending on the sequence of DNA, again on purine sequence of DNA, you get either single or double electron wires. So these electric wires are pretty well characterized. So this is the base pair. And this is a purine containing 2 rings, and these are electron rings, electron pi electron rings, aromatic ones, like in benzene and other aromatic molecules. And this is let's do it blue. That's pyrimidine, and it has 1 pi electron ring. And so these 2 rings produce 2 wires, and this 1 ring produces 1 wire. in every base pair. So it is always 2 on one side and 1 on another, and then they can flip. Now, depending on the sequence. So what we know is that when magnetic field is applied to DNA, then these electrons in the ring start to spin, and then the other way around, when they spin, the magnetic field is produced. Let's do it red. Magnetic field is produced. So then there is an interplay of magnetic field that can go in one direction or another direction. They can flip. Sometimes they can flip one way, another way, and another way, or sometimes that's a hypothesis, but it's pretty confident hypothesis. They can flip this way. So there is a lot of interplay between magnetic fields in the DNA, and that depends on how surrounding magnetic fields are organized. So as you know, in nucleosome there is a magnetic field of nucleosome, possible, also hypothetical. So there is a lot of interference between and play between this merged, unified, fused stack of pi electrons in one strand and 2 stacks of pi electrons in another strand. They all can play magnetically. So these electric wires are pretty well characterized. So this is the base pair. And this is a purine containing 2 rings, and these are electron rings, pi electron rings, aromatic ones, like in benzene and other aromatic molecules. And this is, let's do it blue, that's pyrimidine, and it has 1 pi electron ring. And so these 2 rings produce 2 wires, and this 1 ring produces 1 wire in every base pair. So it is always 2 on one side and 1 on another, and then they can flip. Now, depending on the sequence. So what we know is that when magnetic field is applied to DNA, then these electrons in the ring start to spin, and then the other way around, when they spin, the magnetic field is produced. Let's do it red. Magnetic field is produced. So then there is an interplay of magnetic field that can go in one direction or another direction. They can flip. Sometimes they can flip one way, another way, and another way, or sometimes that's a hypothesis, but it's pretty confident hypothesis. They can flip this way. So there is a lot of interplay between magnetic fields in the DNA, and that depends on how surrounding magnetic fields are organized. So as you know, in nucleosome there is a magnetic field of nucleosome, possible, also hypothetical. So there is a lot of interference between and play between this merged, unified, fused stack of pi electrons in one strand and 2 stacks of pi electrons in another strand. They all can play magnetically. All right. This is the illustration from our paper from 2022, where we illustrate that these electron rings, pi electron rings, oscillate between existence and non-existence. Essentially, they fall apart. They from aromaticity to non-aromaticity. And we illustrate how quantum physical collapse of wave function or a collapse of certainty into uncertainty and back participates in that process. So essentially, we have a few uncertainties here. First, the electrons are delocalized here. Electrons are delocalized. Second, the protons in hydrogen bonds are delocalized. And third, there is an influence of proton wires and surrounding water structures on these protons of the hydrogen bonds. So essentially, this is a base pair, and it oscillates between three states, GC1, GC2, and GC3. And only in one state it is aromatic. Essentially, it participates in electron transfer through DNA. And in other two states, it's not aromatic. And as you can see here, illustration of aromaticity is like 6 pi electrons are merged in a circle and running and spinning together. And here a cat, a proton, grabs one of the two of the electrons from their aromatic ring, and basically the ring disappears. The electrons become localized. From delocalized state, they go to localized state. And in this form, the cat also grabs an electron from aromatic ring, and also the aromaticity disappears. So basically what I show is that these electron wires oscillate very frequently between aromatic states and non-aromatic states, and that is tied up in oscillations with surrounding proton wires and water. Due to limitations of time, I have to refer you to my papers on DNA resonance, and the yellow are highlighted the papers where we use the same principle you saw in this presentation, where we make a molecular model and make a prediction how that would imprint on the genomic sequence, and then use computational genomics to demonstrate that there is some evidence in the genomic sequence and in the genomic data supporting the molecular model. So we use some of the different genomic sequences from human, mice, dolphins, plants, and other species to illustrate and prove our concepts. And also we use functional data like cancer mutations to illustrate the functional importance of proposed hypothesis. This is partial indirect proof, but it still partially supports the proposed models. Although the structure of chromatin is complex, and there are many ways in which it can resonate and form structures with water, there is a way to simplify the answer, to simplify the picture, and this way leads to analyzing the genomic sequence and finding the vibrational code of the genome. So here I summarized the genomic sequence by function. Essentially, there is 5% of the genome, 5% is protein coding. These are protein coding exons. The whole genome is 3.2 giga base pairs. Their blue region is almost 50%, and these are introns. They don't code for proteins, but they code for RNA, which is then spliced out from the transcripts, from the mRNA. And the remaining part is intergenic regions, which usually are not transcribed. So exons work for coding the proteins, and they are unlikely to be involved in resonance signaling. On the other hand, the remaining 95% of the genome are very likely to be involved in the resonance vibrational signaling, and this part is sometimes called junk DNA. And here is the same picture. I only colored here not only introns, exons, and intergenic regions. I also colored which part of that is repetitive. So very small part of protein coding exons is repetitive. It's only 10% of the exons are repetitive. Out of introns, repetitive is about 50% of that, and out of intergenic regions, it's about 60% of this is repetitive. So repetitive sequence is most likely involved in resonance and vibrational signaling, and that's where the answer is to deciphering how the genome produces the morphogenetic field. And lastly, I want to attract your attention to the importance of diffusional heat transfer in DNA resonance and morphogenetic field. This is the area which is rarely discussed. So heat is of great importance for health, and especially for warm-blooded animals. It's important. And its importance is possibly underestimated. So heat exists in two forms. One is radial heat, radial, and another one is diffusional. So in radial heat, it's basically infrared waves. So these are waves, and this is we already discussed in the area of in the slides on electromagnetic signaling. Diffusional heat happens by random movements of water molecules, and the speed of water molecules is gradually transferred through layers of water, and the layers don't have to move around. They can be by themselves, but the heat is transferred just by energy exchange. So first one area is warmed up, and then another area is warmed up, and the heat is transferred this way. So the idea that information can be transferred through diffusional heat came from the book Beyond Weird, Why Everything You Thought You Knew About Quantum Physics is Different by Philip Ball, 2018. Essentially what I learned there is that the information can be transferred between biological objects through diffusion of heat. This information is transferred through quantum entanglement, and it's transferred from, say, from one biological object to another, then to another, then to another. So when the molecules coincide, when they collide, there occurs an information transfer. Even without the waves, through diffusional heat, the information is transferred from one biological object to another. Obviously, this process, the information becomes diffused and messy, but this still can be considered as another way of DNA resonance signaling. So the quantum entanglement happens when the molecules collide, or when the biological objects are connected through radial heat. Heat therapy is widely used in traditional cultures, and it is moksha in traditional Chinese medicine, and there an acupuncture point is pricked with a needle. Needle is inserted, and on top of the needle there is a moxa ball, which is then lit up and burned, and the heat is transferred through the waves, and also it's transferred directly through the needle into the acupuncture point. In many cultures there is a therapy with candles. In Finnish culture there is a sauna therapy, where is natural wood which is used to so people are bathing in radiant heat from natural wood. And pretty much all traditional cultures use bonfire for therapy. Also, there are infrared lamps, far infrared lamps, and far infrared saunas. And there is also traditional therapy where melted wax is used, so that there is, like, say, a joint, and a wax is used to extract their negative vibrations from the joint, and first it is melted, and then it cools down, and as it cools down and solidifies, it extracts the heat and vibrations from the joint, and then this vibration is removed with the solidified wax. So during the process of solidification, some energy, some information is taken out of the joint and brought into the solidified wax. So that's also goes through the diffusion of heat. I would like to speculate that for DNA resonance, it also might be of importance, because DNA is bathed in water, which is warm, and the distance-free path of water, like where water molecules travels freely before colliding with the next water molecule, is very short. It's 0.15 nanometers, and the width of the DNA double helix is 3 nanometers. So the water is very tightly packed in the liquid water and in the nucleoplasm. So the water molecules have very little wiggle, so they don't travel much. So what happens, I believe, is that it's a hypothesis that DNA is colliding, being constantly bombarded by liquid water molecules, and there is quantum entanglement happening. So sequence information from DNA gets into the water molecules, and through the layers of water, it is gradually transmitted through the diffusion to other DNA molecules, and that information, it can be used, and that it can be one of the mechanisms of information transfer, sequence information transfer from one molecule to another. To summarize, I want to highlight the idea that imperfection is the basis of life, and there are continuous, permanently unstable, dynamic self-reorganization processes in nucleoplasm, including the crystallization of polywater, screwing oscillations of DNA in water, and chromatin dynamics. And these continuously unstable states of reorganization are perfectly suitable for subtle effects of the morphogenetic field. And on top of that, there are electromagnetic waves, which are used by nature for signaling between DNA sequences and producing the holographic DNA field. And these include radiant heat and subtle waves, and also acoustic and electroacoustic waves and signaling, and as I just described above, also diffusional heat. As the main goal of the DNA resonance research, I would like to highlight the importance of deciphering the resonance code of the genome. And once the language is deciphered, then we would be able to understand how the morphogenetic field works, and that would allow to manipulate the morphogenetic field and to apply it for organ regeneration, for rejuvenation, for treatment of multiple diseases. Especially that would be helpful for understanding how the mind works and consciousness, since I believe mind and consciousness also are functioning, interfacing with the body through the DNA resonance mechanisms. So this would bring a lot of applications for medicine and therapy. Additionally, I think very importantly is to develop vibrational literacy, vibrational ecology, and avoid the collapse of the collective morphogenetic field and biofield of the humanity. Currently, we are facing increased attempts of genetically re-engineering humans and animals and plants, and that is done without the knowledge of vibrational genetic code and vibrational genetic language of the genome. So by understanding how that works, we would be able to avoid damaging the biofield and morphogenetic field, individual and collective. In conclusion, I would like to thank people who understand all the details of my work, which are my main co-author Ivan Savelyev, also Oksana Poleskaya and Michael Rempel, and my close people who co-authors who discuss with me the questions and discuss with me the science, Nelly Zырянова, Alena Naumova, and Richard Alan Miller, and also Alexander Vikharev, who did the programming for this project, and in Plastun Zhilidin and Filin, who did modeling of the polywater in 3D, and my past collaborators and workers. The work was largely funded by me and partly crowdfunded, thanks to the donors. And you can find all the full text publications and some of the videos on the website dnaresonance.org. And if you like to contribute, this is our crowdfunding information, and there's are my contacts. I welcome voice messages on WhatsApp and Telegram, and obviously email is good as well. Thank you very much, and I would welcome any feedback. Good day.